Thank you, Zeke, for the nice introduction, and thank you, Sabine, for the invitation to this nice symposium. So let's see, not proceeding to next slide. So these are my disclosure. And here is the Carrier Lab. So we present you data obtained at the Institute of Experimental Pharmacology and Toxicology under the supervision of Lucie Carrier, a wonderful scientist. And the group is interested in elucidating the genetics, pathophysiology, and therapy of inherited cardiomyopathy. This nice picture has been taken in summer 2019, long before the pandemic, and we're all still smiling. So in this talk, I would like to give you some insight on MYBPC3 and hypertrophic cardiomyopathy, on molecular mechanism, and finally on gene therapy. So we have heard already all the day long actually about HCM and the features, and therefore I don't want to bother you now at the end of the day, but just again, HCM has a left ventricular hypertrophy with a septal thickness increased, and at the cellular level we have myofibrillar disarray and a lot of fibrosis. And we have also heard that there is an heterogeneity of genes in which mutation are linked to the disease. And the most affected one are MYBPC3 and MYH7. And we heard from Perry Elliott that the first gene discovered was MYH7, but actually forgot to mention that back then in early 90s, also MYBPC3 has been discovered exactly in the same issue of Nature Genetic by Lucie Carrier in the group of Kathy Schwartz. So here is an image of adult mouse cardiomyocytes with a nice immunofluorescence staining for alpha-actinin in green and the cardiac myosin binding protein C in red. And this is to show you the sarcomere, which is the unit, the contractile unit of the sarcomere. And you see that the cardiac myosin binding protein C is transversally located in the C zone of the A band. And this protein is encoded, is present on the chromosome 11, and the gene is about 20 kilobase per long and encoded by 35 exons. The protein, you see here, the structure comprised 9 immunoglobulin domain and 3 fibronectin-like domain. In yellow, highlighted in yellow, you can see the cardiac-specific regions, which makes a difference with the slow and fast skeletal isoform of the myosin-binding protein C. This protein has many interacting partners on the thin filament, with the actin on the end part of the protein, and with the myosin on the N-terminus, but also on the C-terminus. The C-terminus of the protein is also very important for incorporation of the protein into the sarcomere. There are many post-translational modifications which regulates this protein. And the role of this protein is very important because it is responsible of regulation for the relaxation. So it binds to the myosin and pull it back to the rod. So there is this super relaxed state of the myosin where the myosin head cannot bind to the actin, and therefore the relaxation during the diastole can be completed. You see here again the exonic structure of MYBPC3, and in red and blue there are all the mutation which has been described in the literature. And interestingly, for this protein, for this gene, over 75% of the mutation are truncating, which means that they produce a shortened protein. And there are also some in-frame deletion or insertion deletions. Mainly, some of these truncation are so-called founder mutation, because in a certain population or geographical area, they are found very often. For example, a mutation here in producing frame shifts and the truncation in the C2 domain is responsible of 57% of all HCM cases described in Iceland. There are also many others founder mutation in the Netherlands. For example, there are many described, but also in Italy, and the place where I come in Tuscany, this mutation is quite well described. And this is actually the mutation we introduce into the mouse to have a MYBPC3 targeted knock-in mouse to have to mimic the situation in the human. So normally the HCM patient carry heterozygous mutation, which have a later onset of the disease and maybe a milder progression. However, there are some cases of B allelic mutation described in the literature, and particularly the B allelic truncated mutation are correlated with a very severe phenotype and a very early onset. You see here that the mean age at the diagnosis is only 37 days, and that the mean age at death is 97 days. So these babies are brought from the parent to the hospital because of poor feeding, of excessive sweating, of bad respiratory situation. And when they are examined, the physician can find right away a very remodeled heart, which can present a sign of hypertrophic cardiomyopathy, but also of dilated cardiomyopathy and of left ventricular non-compaction. For these kids, there's no, I mean, we can treat them pharmacologically, which is only a symptom treatment, so it's not curative. And these kids are dying very early, before 1 year of age, because of heart failure. And the only way to save them is the heart transplantation. And these kids have B allelic truncating mutation. When we compare with some other cases described in the literature with the MYBPC3 truncating missense or missense mutation, we see that the age at diagnosis is higher. The mean age is at 20 years. So for these kids who has a truncating mutation in MYBPC3, there is no treatment other than heart transplantation, and there's no, there is a very low or absence of MYBPC protein. So what is the molecular mechanism behind this? So how is the expression of a truncating mutation regulated? So we have here a situation of a monoallelic truncating MYBPC3 mutation. So we have a truncating allele, which will produce a nonsense RNA, which is then regulated by the nonsense mediated RNA decay, which reduce the amount of these mutant mRNAs. And in case something is translated, the UPS will provide to eliminate all this truncated protein. The wild type allele, in contrast, will produce a normal protein and will try also to compensate for the absence of C3 protein. But at the end, we will have an upload insufficiency, and this will bring to a hypertrophic cardiomyopathy. In the case of a patient carrying a B allelic truncating mutation, both alleles are under the control, let's say, of NMD and UPS. So at the end, there is really no protein at all, and this results in an even more severe phenotype with heart failure and death before 1 year of age. So in Hamburg, we were collaborating with the Heart Center, and we obtained a skin biopsy at the time of septal myectomy from an heterozygous MYBPC3 patient having HCM, of course. So we reprogrammed the fibroblasts and generate human-induced pluripotent stem cells, which were then used together with the CRISPR-Cas9 to create a nasogenic control and to mimic the situation of the very severe phenotype in the infant with the B allelic truncating mutation. We introduced even a second mutation. We then did the cardiac differentiation. You see here a monolayer of cardiomyocytes beating, which were then used in 2D as 2D cardiomyocytes or as engineered heart tissue to reveal phenotype, and then possibly to test a therapy and then make a therapeutic decision. So in a very recently published paper in stem cell research, we then published this cell line. So this was the patient-specific cell line, the isogenic control in which we repaired the mutation, and the compound heterozygous cell line carry a second mutation in exon 27 at the homozygous state, and we introduced one of the founder mutation found in the Netherlands. We did several quality controls of this cell line. There was no cardiac, no karyotypic abnormalities, no off-target associated with the genome editing, and these three cell lines were all able to differentiate in all three germ layers, and particularly we were interested in cardiomyocytes. So we did want to verify whether these truncating mutations are really leading at the absence of C protein. And therefore, we just performed Western blot with protein lysate from cardiomyocytes cultured in monolayer. And we could observe that the heterozygous had only 50% of wild type C protein compared to the isogenic control. And in the compound heterozygous, we could not even find anything. And we did the full blot also to prove that there was no truncated protein revealed. So this is really missing protein, the problem. So of course, we can imagine of different strategy to correct now the mutation and this problem of absence of C protein. So you see here again the scheme of MYBPC3 with a different mutation, and we can imagine several levels of therapy. So with the CRISPR-Cas9 therapy, we could imagine to tailor each single mutation with a guide RNA and repair the mutation. This is, of course, very difficult to go through the FDA and accept this as a drug for each single mutation. So in another level, we could use antisense oligonucleotide for exon skipping. So this works about only for the exons which can be skipped in frame, which means that the protein which is resulting is an in-frame protein but shorter, and this should be still functional. So you see here some example. We could end up by using this several antisense oligonucleotide, we could remove 35 protein mutation. In another level, we could use transplicing. So here we can imagine to have two molecules. So it's much easier to go to the FDA with this. Two molecules to remove all the mutation at the N terminus or all the mutation at the C terminus of C protein. And transplicing works, you have practically the wild-type cDNA on one side and the binding site for the pre-mRNA endogenous. And through the transplicing, you will have a wild-type full-length cDNA. So we tested this in a mouse model, in our knock-in mouse model, and it worked in principle, but the efficiency was so low that the protein produced, the amount of protein produced was not enough to overcome the phenotype. In the last strategy, we can think of a gene replacement. So this is the more elegant strategy in which we take the full-length cDNA of the missing gene, and we can introduce this with a vector, for example, in the cardiomyocytes, and then via transcription and translation of the full protein, we will have a replacement of the missing protein. So we use this gene replacement in the MOBPC3 knock-in mouse model. We did the study with a dose dependence of several doses of an AV9 carrying the flag-tagged murine MOBPC3 sequence under the control of a TNT2 promoter. And we observed dose-dependent increase of mRNA wild type and of protein. And this was going together with the reduction of dysfunction and of left ventricular hypertrophy. We did also tested in engineered heart tissue from murine knock-in and knockout whether this was possible, and indeed we could observe also increase in mRNA protein of the wild type C3 protein together with a reduction of a dysfunction. In a IPS model, these were patient derived with a truncating heterozygous mutation. We tested the particular cellular type of AV9 with a human construct flag-tagged, and we observed increase in mRNA and protein, and this was leading to a reduction in cardiomyocytes hypertrophy as reduction of cell size. So in summary, and this is almost my last slide, I showed you MOBPC3 as one of the most affected genes in HCM, that most of the MOBPC3 mutations are truncating, resulting in the absence of protein, haploinsufficiency or deficiency. That gene replacement for MOBPC3 has been successful in mouse engineered heart tissue and in human iPS-derived cardiomyocytes. And we firmly believe that the gene replacement therapy would be an option to treat this infant with a severe form of HCM due to BLL truncating mutation. And with this, I would like to thank all my previous colleague at the Institute of Pharmacology and at the UKE Heart Center, Julia Milch and Monica Patton, and some other collaborators. And now I left the UKE to go to Dynacor, a new company which is translating this concept of gene therapy from preclinical to clinical asset. And with this, I would like to thank you.
Thank you, Julia. Thank you a lot for this very interesting talk. There are no questions from the audience, so I take the liberty to come up with questions on myself. What would be your estimate? You had on one of your figures that approximately 30 cases, of this double truncating C3 mutations. What would be your estimate? How many cases are around there in Europe or in Germany? So what is the incidence of this?
Okay, so I can give you a rough number. We estimate due to the prevalence of MOBPC3 mutation and the biallelic, the truncating mutation, we thought that we can have about 700 births worldwide per year that we could target.
That's a high number. And one more question, reflecting your mouse models. So in the mouse, you could show already that having this gene replacement therapy would definitely increase the cardiac, insufficiency, would reduce cardiac insufficiency and increase the cardiac function. Is there a particular time window? So do you have an indication that it would be important to have as early the treatment as possible, or would it be also sufficient to have it as a several-week-old infant?
Okay, so the slide I show you was referring to a prevention study. So we did the injection before the phenotype was occurring, so we were able to prevent the development of the phenotype. In a second study, we try to rescue the phenotype, but we end up with a bad quality of virus preparation, and we could not. We had n equal 2, and we could not produce as much protein as we knew we need from the EHT experiment to revert the phenotype. So it was just a matter of academia production. So I'm sure that we will be able to rescue the phenotype, and therefore also think of a gene therapy for a heterozygous patient.